students, this video deals with structure and functions of mitochondria. Introduction to mitochondria. It is the powerhouse of the cell. It is the second largest organelle in a cell. It is a main source of adenosine triphosphate, the energy-rich compound that drives the fundamental cell functions. Its functions include force generation in muscle contraction, cell division, biosynthesis, folding and degradation of proteins, generation and maintenance of membrane potentials. So these are abundantly found on the sites where the energy is earnestly required, such as the sperm tail, muscle cell, liver cell. Up to 1600 mitochondria are present in the actively dividing cells and also in microvillus. ATP is produced on a massive scale in the human body, amounting to 50 kg per day in a healthy adult. Mitochondria are oxygen-consuming organelles of immense importance, floating freely throughout the cell. The enzymatic oxidation of chemical compounds in a mitochondria releases energy. Typically, there are about 2000 mitochondria per cell, representing about 25% of the cell volume. In 1890, the mitochondria were first described by Richard Altman, and he called them as bioblast, and Bender in the year 1897 coined the term Mitochondrion. So this is the picture of mitochondria, where you can see the outer membrane, inner membrane, and its internal matrix. Coming to the structure of mitochondria, we have the outer covering. It is called the outer membrane. The organelle is enclosed by two membranes, the outer membrane and inner membrane. The outer membrane resembles more than it is similar to the plasma membrane in structure and chemical composition. The outer membrane constitutes as a semi-permeable phospholipid bilayer made up of porins. The porins are nothing but protein molecules which helps in the transport. So this layer is permeable to ions, ATP, ADP and nutrient molecules. Porins in the outer membrane allows small molecules to be exchanged between the cytoplasm and the inside the mitochondria, that is intermembrane space. So this is, I have shown you the outer membrane of the mitochondria. Here you can see the inner membrane, it is folded. Inner membrane of mitochondria, it is rich in many enzymes, coenzymes and other components of electron transport chain. It also contains proton pumps and many permease proteins for the transport of various molecules such as citrates, ADP, phosphate and ATP, where these are the components in citric acid cycle. The inner membrane is a complex but permeable membrane made up of complex molecules of electron transport system, ATP synthetase complex and transport proteins. This layer allows oxygen, water and carbon dioxide. The inner mitochondrial membrane, it forms a finger-like outgrowths or projection, we call them as cristae, towards the lumen of the mitochondria and contains a tennis rocket shaped F1 particles that contain ATPase enzyme for ATP synthesis. Numerous transporters in the inner membrane ensures that import and export of important metabolites and markedly folded into tubular inner mitochondrial membrane, so which has a large surface area at which encloses the matrix space. So this is the inner mitochondrial membrane. It is invaginated to form a finger-like projection, so just to increase the surface area for the metabolic reactions. Intramembrane space. In between the outer and the inner membrane of mitochondria, it has same composition as that of the cell cytoplasm. It is primarily responsible for oxidative phosphorylation, and there is a difference in the protein content in the intermembrane space. So this is the region in between the two membranes. So this region is called intermembrane space, and here it is, this region is called a matrix. Mitochondrial matrix. The mitochondrial matrix, which is a liquid colloidal area encircled by the inner membrane, contains the soluble enzymes of Krebs cycle, which completely oxidize acetyl-CoA to produce carbon dioxide and water and hydrogen ions. Hydrogen ions reduce the molecules NAD and FAD, both of which pass on the hydrogen ions to respiratory or electron transport chain, where oxidative phosphorylation takes place to generate ATP-rich molecules. That is, to generate the energy-rich molecules, that is ATP. The matrix has a single or double circular and double-stranded DNA molecule called mitochondrial DNA, and it has a 55S ribosome called mitoribosomes. Since mitochondria can synthesize 10% of the proteins for their own function, the protein synthesis, it has the ribosome, that is protein synthesis machinery. They are considered as semi-autonomous organelles. So this is the matrix of mitochondria, which is rich in metabolic enzymes. Coming to the functions of mitochondria, the most important function of mitochondria is to produce energy. Mitochondria produces the molecule adenosine triphosphate. It is called energy currency of the cell, that provides energy to drive the host cellular reactions and mechanism, that is metabolic reactions. The simpler molecules of nutrition are sent to mitochondria to be processed and to produce a charged molecules, and these charged molecules combines with oxygen to produce ATP molecule. This process is known as oxidative phosphorylation. The function, so I have listed the function of mitochondria. The first function is the TCS cycle, the production of energy. After glycolysis, the primary mitochondrial function in animal cell is production of energy in the form of ATP and the regulation of cellular metabolism. So after glycolysis, which takes place, so glycolysis takes place in the cytoplasm, and TCS cycle, which takes place in the inside the mitochondria. So this is the pathway I showed you, the glycolysis, where the glucose is degraded into 2 molecules of pyruvate. So this is a schematic representation of Krebs cycle. The reactions are involved in the production of ATP are collectively known as citric acid cycle or Krebs cycle. The food is broken down into component parts by the process of metabolism. The food molecules are transferred to mitochondria, that is glucose after completely degraded into pyruvate. The mitochondria, where they undergo further processing through the Krebs cycle to produce energy. After a number of complex reactions in the presence of oxygen, the oxidation of fat, carbohydrates takes place in the Krebs cycle. It is a site of important reaction that produces the energy for all cellular reactions. So this is called a central metabolic pathway, which produce the intermediates for all the reactions which takes place inside the cell. The next important function, it is the electron transport chain. The inner mitochondrial membrane, it has a complexes. The transfer of electrons down to the electron transport chain is responsible for energy production. The inner membrane mitochondria is a site of this electron transport chain. So this electron transport chain, it has a different complexes, that is NADH, FADH, then you have cytochrome C and ubiquinons. All these have a role in the process of electron transport across the inner membrane of mitochondria and results in electrochemical proton gradient. This proton gradient is responsible for the synthesis of ATP. See, after Krebs cycle, this that is the products, that is FADH2, NADH2, which are produced. So this proton pumping takes place across the inner mitochondrial membrane. This is the complex 1, NADH reductase, complex 2, succinate dehydrogenase, complex 3, it is cytochrome oxidase, that is cytochrome BC, and the last one is cytochrome oxidase, where the proton pumping takes place. So because of the proton gradient, so where there is So when there is a release of energy because of proton gradient, there is a phosphorylation of ADP into ATP. Thus the synthesis of ATP takes place in a ATP synthase, a complex which is present in the inner mitochondrial membrane. The third function, it is ATP synthesis. As I told you earlier, the synthesis of ATP is an important mitochondrial function. So this is the structure of ATP synthase. You have a different subunits, that is alpha, beta, gamma, delta, all these products. It is a lollipop-like structure. It produces the products obtained from Krebs cycle, and the amino acid oxidation or fatty acid metabolism they are used in the synthesis of ATP. The proton gradient created by the electron transport chain drives the process of ATP formation in the mitochondrial matrix, so which is responsible for all the other activities which requires energy. The fourth function of mitochondria, it does the function of apoptosis. It is also called programmed cell death. The programmed cell death is carried out by an organism in the developmental stage where there is an unwanted cells that need to be destroyed. Apoptosis that enables the digits of the hand to separate into fingers during the fetal development. It is also carried out in the when the whole body detects the infection or is an immune system responses to the bacteria or an illness. Since the oxygen is required for the production of ATP, in the condition where the body is combating with an illness, the consumption of oxygen rises. As a result, which the certain mitochondrial reactions cause the production of free radicals. These free radicals in excess of mitochondrial ability to detoxify the cell and causes the damage to the mitochondria, and the abnormal cell death due to the malfunction can give rise to the medicinal conditions as interface with organ function. That is, the normal cells, when there is a illness or during the programmed cell death, the cell shrinkage takes place because of chromatin condensation, and this condition is called membrane blubbing, and the nuclear collapse takes place, and the nuclear collapse and continued blubbing leads to the death of the cell. That is called the lysis of apoptotic bodies. That is called apoptotic body formation. This whole process is called as programmed cell death. The fifth function I have mentioned here, it is the urea cycle. So urea cycle, it has the 5 reactions. Out of 5 steps, the step 3, that is the formation of arginosuccinate, it takes place in the mitochondrial matrix. So urea cycle, it is a mitochondria is also a function of mitochondria. So that is, the mitochondria is a site of urea cycle. The last function I have mentioned here, the mitochondria produces the heat and accumulate iron containing pigment, the heme ferritin ions of calcium and that is phosphate. The example in the osteoblasts of bones and yolk proteins. Mitochondria help the cells to maintain the proper concentration of calcium ions within the compartments of the cell. The calcium entry into the mitochondria plays an important role in cellular calcium homeostasis, energy production, and mitochondrial health. So this is overall function of mitochondria, structure and functions of mitochondria. Even though mitochondria and their membrane protein complexes have been studied intensively for more than 5 decades, they remain a constant source of fascinating organelles and unexpected new insights. So it's an open question about many of them of a fundamental nature and of direct relevance to human health. So this is the link I have given you you can view the 3D structure of mitochondria by clicking this link. Thank you for watching. Thank you.